So we've already talked about T follicular helper cells, subscapular sinus macrophages, all that stuff, right? We've already talked about somatic hypermutation. We've already talked about isotype switching, but I want to just take all the stuff that we've learned about kind of over the past couple of weeks and collectively put it together into what is happening in the secondary lymphoid tissue, right? So naive B cells are going to enter the lymph nodes through the same mechanism as the T cells, right? Through the high endothelial vanules, the receptor 7 interacting with the ligand 21 and 19, and then they're going to migrate to the B cell zones through the receptor 5 and the ligand 13, and then they're going to be encountering the specific antigen on the follicular dendritic cells of the subscapular sinus macrophages to become activated, right? So the antigen, that's the first signal, and then the second signal is usually, like I said, from T follicular helper cells, especially in the context of the lymph nodes. So some of the activated B cells are going to move to the medullary chords to form a primary focus of dividing B lymphoblasts and become plasma cells. And the reason that we're doing this is because we're able to supply an early source of IgM, right? So it's not as bad as what we saw with the B1 cells, but it's a little bit more precise than that, but not nearly as precise as what you get after all the affinity maturation that B cells are going to undergo. So some of the B cells are going to migrate back to the primary follicle, initiating a germinal center reaction that forms the secondary focus of dividing lymphoblasts. Well, if you've ever gotten a infection, say, I don't know, the flu, everybody gets the flu, I think, at some point in your life, you end up getting swollen lymph nodes. And the reason that you're getting swollen lymph nodes, and the reason why the flu can take almost a week, really, for you to get over is because as you're getting those swollen lymph nodes, you're getting a highly specific response to that specific pathogen, right? So what's happening is we have affinity maturation. Now remember, that's what happens with the whole somatic hypermutation, kind of the Darwinian selection on steroids, if you'd like to think about. Class switching, isotype switching, those are the same things. And then generation of plasma cells and memory cells in case the pathogens should come back. So this diagram here is really nice. It's helpful, but pedagogically, I don't find it to be that great. This is, I think, from fluorescent antibody staining. And then down here we see, I think, a light micrograph and then germinal center that's been stained showing the follicular dendritic cells. I don't really find either one of these guys helpful, but what I do find helpful is this picture right here. So here we see the dark zone, the light zone, and then up here is the mantle zone. Okay, so in the dark zone, what we have is centroblasts, remembering your, I guess, Greek terminology from general biology. And then in the light zone, we have follicular dendritic cells interacting with the centroblasts, and then kind of also centrocytes as well. I think more so centrocytes than centroblasts. And then in the mantle zone, we have fully mutated B cells, fully developed, if you can think about it, B cells interacting with T follicular helper cells. Centroblasts are kind of like, this is kind of new terminology. Remember your stuff from your blast versus your site. These are just B cells, right? Don't let that terminology throw you off. So follicular dendritic cells produce interleukin 6, 15, and BAFF, and this is really the thing that these follicular dendritic cells are kind of known for, is the rapid division of B cells. And one of the things that I don't know if I've stressed this enough, but this interaction with CD40 with CD40 ligand is absolutely essential for many, many, many reactions in immunology. And in this context, interaction inducing the B cells to undergo somatic hypermutation, but also AID plays a role in isotype switching. For this differentiation from centroblasts to centrocytes, the actual mechanism for it is poorly understood, and I was kind of disappointed by that. But it almost kind of reminds me of what we saw with in the bone cells, the osteoclasts to the osteoblasts, to osteocytes. But I don't really know much details on that, whether it's a contact thing or through cytokine interactions. But anyways, centrocytes are going to be dividing much more slowly. They're going to start expressing their surface immunoglobulin. They're going to undergo competition for attention by both the follicular dendritic cells and the CD40 ligand. This is kind of collectively what we talked about with affinity maturation. Now, if they do not get interaction with both the CD40 and CD40 ligand, so by extension attention from the T follicular helper cells or attention from the follicular dendritic cells, they need both of those things. If they don't get that, they die. And this is where we're getting that really highly selective process going on here. For isotype switching, these are the cytokines that are involved in this. This picture is great. This picture is awesome. I'm not really going to stress the specific identities of the cytokines that are involved in isotype switching. We've already talked about the mechanism behind that, and if you understand the mechanism, that's okay, because you don't really need to know the specific identities of each and every player, but it is interesting to look at each and every one of these. Plasma cells and then memory cells, the only thing that I really didn't get to mention in the, I think my first video, was the fact that when you're talking about differentiation into plasma cells, and let me switch to a color that's a bit more visible, plasma cells, the main cytokine is interleukin 10, and when we're talking about memory cells, the main cytokine is interleukin 4. But both of these, in order for the B cell to be receptive to either one of these, it has to have CD40 and CD40 ligand interaction. And obviously, I didn't mention it, but the B cell receptor is going to go plays a role in receptor-mediated endocytosis, antigen processing and presentation, presenting a specific peptide MHC complex for the T follicular helper cell to interact. And if that interaction fails, then none of this other stuff is going to happen. Don't miss the forest for the trees here. Really cool kind of, I guess, comparison and contrast of the naive B cell with the plasma cell. This is great. You should study this. I'm not really, I can't really talk about this though. 3 areas that's happening. We have the T cell area, the medullary chords, and then the primary follicle. Inside the medullary chords, we have the primary focus. One of the things that I was going to say about this is that we have dividing B cells and dividing T cells. One of the events that we have happening also in the medullary chords is very, very fast responses of differentiating B cells into plasma cells that are going to be making IgM antibodies. The B cells differentiating into plasma cells, this is really just the IgM type. Remember, quality versus quantity. And the T follicular helper cells are going to be secreting interleukin 5 and 6, and that's going to result in the production of a transcription factor known as B lymphocyte-induced maturation protein 1, or BLIMP1. And I'm just going to write TF because it's a transcription factor. In the primary follicle, we have B cells, and then to a lesser extent T cells are still actively proliferating, and this gives rise to something known as a germinal center. This germinal center consists of B cells, T follicular helper cells, and then follicular dendritic cells. Ultimately, we have B cells differentiating into plasma cells and memory cells, and this is happening in various stages of this giant spherical like structure, right? So the first thing that happens is B cells are going to kind of start to differentiate into centroblasts. Things that cause this differentiation, well, both the follicular dendritic cells and the T follicular helper cells are both secreting their own cytokine. I don't know how well you can see that. Cytokines. What are the cytokines of the T follicular helper cells going to do? Well, they're going to cause basically the downgraded production of the B cell receptor, and we're going to start somatic hypermutation, and if needed, isotype switching. Decrease in B cell receptor production, which leads to AID, which plays a role in both isotype switching and then somatic hypermutation. A hypermut for hypermutation, and then isotype switch. Or I'm going to say iso switch for that. Now the cytokines of the follicular dendritic cells, all that these guys are really going to do is these guys are going to promote further, I guess, cellular division and cellular proliferation. See that? Cell proliferation. Maybe I could just say it like 3 times. The follicular dendritic cells secrete cytokines, which predominantly causes cellular proliferation. Next thing that happens, and the mechanism for it is not quite well understood, is that centroblasts are going to differentiate into centrocytes. Cytes are then going to kind of compete for survival through kind of the affinity maturation that we said that requires both the attention by follicular dendritic cells, and by extension I mean antigens, and the T follicular helper cells CD40 ligand. And I don't really have room to kind of, I guess, fill it out, but I guess I'll try to stretch this out here. Centrocytes are ultimately going to end up differentiating into plasma cells and then memory cells. The follicular helper cells are all about, I guess, their cytokines that are playing a role in this. And in this context, it's interleukin 10 for the plasma cells and interleukin 4 for the memory cells. For the memory cells, there's really not much more I could say about that. But as for the plasma cells, there's ones that are going to kind of stay in the lymph node, and they're going to start to secrete kind of soluble cytokines that are going to be carried away through the lymphatic system and ultimately end up at the site of the infection. These guys have a pretty short lifespan, but for the ones that are going to actually migrate to the bone marrow, and I'll talk about why they're migrating to the bone marrow later, these ones have a pretty long life cycle. They stay in the secondary lymph, and then we have the ones that go to the bone marrow. I don't really have room to write it out, so I'm just going to say it twice. For the ones that stay in the lymph, they're going to be secreting soluble, as you can imagine, Igs or antibodies. These are going to be carried away to the site of the infection. These are relatively short-lived. Why are they short-lived? Because we have lots of stuff moving in and out of the lymph node. But as for the ones that are going to migrate to the bone marrow, the main reason why they do this, and hopefully I can just kind of fit it in here, and imagine especially if they were, say, IgE antibodies, for example, something for an immune allergic response. IgE antibodies need to interact with basophils, mesophils, whatever. Or say something that was going to act on neutrophils or something else, depending on the specific identity of the antibody. We haven't really talked a whole lot about antibody effector functions, but you're at least relatively familiarized with that. The reason why we go to the bone marrow is because that's the site where the mast cells, the basophils, the eosinophils, all of that stuff, that's where we can find all of it. So it's in direct proximity to the cells that it's going to interact with. So I'm just going to say near other cells.